Hi, my name's Pavlin. This is really joint work with my collaborators at the University of Ljubljana. Like that? Better? Okay. Okay. Good. So I'm just going to read the title, which is Teaching Bioinformatics Through the Analysis of SARS-CoV-2 Project-Based Training for Computer Science Students. I think that's very self-explanatory, right? So what I'll really be talking about is how we designed a course that's an introductory course for bioinformatics around this project-based setting, right? So I think it makes sense to sort of contextualize the course itself. So we're from the Faculty of Computer Science. This is an elective course for master's students. Our university or faculty doesn't really have very much to do with bioinformatics at all. So most students don't really have any like biological background, right? We have a lot of machine learning courses or programming courses, right? But this is the only bioinformatics related course at our university or at our faculty. It's also an elective course, so it's not a lot of students. I mean, the students don't have to take it. It's a quite popular course, but it's elective, right? The course itself is made of lab work and a final exam, and really I'll be talking about the lab work. The lab work is made of 5 homework assignments that the students need to do, and if they pass the homework assignments, they can then go to the final exam. And every assignment has around 4 mandatory problems and then some optional bonus problems for extra credit, right? Since we're the Faculty of Computer Science, we expect a strong baseline in programming, right? So,
Sorry, you're not sharing the screen.
Oh,
the other one.
That one.
We are pausing your session.
Yeah.
For.
All right.
The guys online are not seeing anything. That's why.
I knew it.
Yeah. Couldn't be so easy.
That's what I was sharing.
Yes.
I knew it.
And then if you do this. Yeah.
And we are back from technical difficulties.
Okay.
I can see it. It's fine.
Okay. All right, sure. It's fine. Okay.
Getting better. Just the camera. Seems we're talking about a camera. No, we're good. It's fine. Yeah.
Is it okay? Okay. Wonderful. Okay, so as I was saying, we have computer science students, so we expect a high level of programming ability. We can expect that, hopefully. And so we use Python, and we found that Jupyter notebooks are really nice for carrying out these homework assignments. For the actual logistics, we also found that GitHub Classroom is really nice. So GitHub Classroom is this platform where you just create a template repository with all the instructions. And then every student clicks a link, and then they get their own home GitHub repository, and then it's just a private one, so only they can see it. But of course, you can see it as well. And it makes it really nice for grading. So we found GitHub Classroom to be very nice. So the course, of course, consists of lectures, and the professors, they introduce the theoretical concepts, they talk about the algorithms and the background, and then the lab work, right, are really meant to solidify this theoretical knowledge. So they implement the algorithms that they heard about, and then very much apply them to like a real world case. And now the key thing here is that we focused all of this lab work around the COVID. Right. So a few years ago, when we started running this course, of course, it's very unfortunate that COVID happened, but the silver lining is that we were able to make these nice exercises, right? Because it really highly motivated the students, right? Because they were seeing stuff on the news about it. And then here we had them make the same analyses as the real scientists were doing, right? Because some of the algorithms are really not that complicated, right? And they were able to reproduce a lot of the stuff, and then they were very happy when they say, now I know why it says this on the news, right? So it was really very motivating for them. Maybe not anymore now since the COVID is gone, but, so very briefly, right? So the course structure, in Slovenia, we have 2 semesters. I know some universities have it different. It's around 14 weeks, so we have plenty of time. We space out the assignments around about equally, right? So it spans the whole semester. And there's plenty of space for lectures that are sort of orthogonal to the assignments, right? So for instance, we have a lecture on genome assembly, but they don't have to, the students don't have anything to do with the genome assembly because implementing that is quite difficult. We tried, but it didn't work out too well. Okay, so next I'll just go over the assignments quite briefly because this really is the meat of the thing, right? So in assignment 1, right, the students really get a first feel for the genome and the bioinformatics sort of data, right? So we literally just give them a FASTA file with the SARS-CoV-2 genome and then say, okay, now you have to do some things. And this is a bit of a shock for them. So we make the exercises here a bit simpler. Okay, so we have this long DNA, right? And then what's the first thing we want to do, right? We want to find like maybe some coding stuff, maybe the regions that actually do things. And so we're going to try to find genes. And all the genes are sort of like encapsulated in these open reading frames, which is very simple to find. You just find a start and stop codon, and anything in between is an open reading frame. So they have to implement this very simple algorithm. And then once they have it, they have to apply it to the SARS-CoV-2 genome, right? Okay, now what, right? So now you have these open reading frames, and next we're going to try to figure out what they do. And we take a very simple approach here. So what we do is we just translate the open reading frames to proteins, and then we calculate the average hydrophobicity of the amino acids, right? And what's really nice about this is because this is a very simple way to identify transmembrane proteins, right? So we're not doing like full functional annotation here, but it's a very simple thing where we can get from a genome to genes to figuring what they do. So this is the first exercise. And as a bonus exercise, they can also create a visualization of these ORFs that they find. And then they can make a nice visualization like this. The projector is sort of graying it out. But it's very nice to see because you can see all these open reading frames where they start, and then you have an abrupt stop with the stop codon. It's also interesting here that almost all of the open reading frames are on the positive strand, like on the top of the black line, because SARS-CoV-2 is a single-strand RNA virus, right? And you don't really see much on the negative strand because it isn't there. Okay. Then we move on to the second assignment, which is really more of like a functional annotation, right? So usually the way we would do this is with BLAST. Of course, BLAST is very complicated, and we're not going to have the students code a BLAST. But we do something similar, right? So this whole exercise is sort of centered around alignment algorithms. And we sort of mimic BLAST. So the first step of BLAST is so you construct this sort of database. Here, what we do is we give them 20 genomic sequences of related viruses, and then they have to find the 3 closest viruses, the most related viruses, and then they construct their own database of these annotated genes. So they have to implement this global alignment algorithm and then apply it to the SARS-CoV-2 genome and find the 3 most closely related viruses, and then they have to build up like this database. Then when they actually annotate things, right, we have these ORFs from the first assignment, and now we're going to figure out what they do by running like the local alignment, sort of like BLAST does, to the database. So what they have to do here is just implement this local alignment algorithm, which is very similar to global alignment, and then query it against the database and see what does what. Right? As a bonus exercise, this is quite an interesting one. I like this one a lot. We showed them that these alignment algorithms are actually quite flexible, right? So the SARS-CoV virus was the 2002 virus. It's related to SARS-CoV-2. And they found that it has a particular drug binding motif that was used for medicine, right? And then we have them see, can you do this on the SARS-CoV-2 virus? Can you find a similar binding motif? And by designing this sort of clever scoring function, you actually can do that. Okay, so now that we figured out what the genes do, we sort of move away from that a little bit, and then we go to phylogeny, right? So as we heard in the keynote this morning, right, the phylogenetic inference can be very complicated. Of course, we're not going to have the students implement these complicated methods, but we just go with the neighbor joining, right? Neighbor joining is a very simple algorithm that you can get done in about 30 minutes, and then we have them implement routing, and then also the drawing, right? And then they just apply this to some coronavirus sequences that we provide, right? And then if they apply it to these, for instance, 31 of these sequences, they get nice little phylogenetic trees like this, which is quite nice, right? Evolution is not always top-down and straightforward like that. So we also have them sort of look into recombination. So for a time in the news, they were talking about that SARS-CoV-2, did it come from bats or did it come from pangolins at some point? And we have the students follow this study from Lamital who did this. And the approaches they use are very simple, so the students can do this very quickly. But they really see that it's not just like evolution top-down, but it's a bit more complicated. And as a bonus exercise, we also have them implement this tree bootstrapping, just so they know how to assign confidences to the trees. Okay, so the next one, in the next assignment, we look at viral evolution, and we look at variants. Here, we give them the NCBI sequence, and then we give them 211 viral sequences from Slovenian COVID variants. And their goal really is to figure out how fast is the virus mutating, and we can do this by calculating the mutations per nucleotide per day, and then we compare this against other viruses. So this is one of the parts. And the other part is to classify some of these SARS-CoV-2 genomes into the variants, right? So the variants are very clearly characterized by known mutations, and we have them develop some classification scheme, and then in the end, they're able to generate a figure like this, which shows the prevalence of different variants in Slovenia through time, right? And we see this alpha, and then delta, and omicron, and this is very similar to what you see on the official online resources, right? Okay. In the final last assignment, we sort of move away from this. We sort of move away from the DNA, right? Because all of them until now have been really working with the strings, right? With the DNA string. Here we look at gene expression data, right? So a large part of bioinformatics is also related to gene expression, and this sort of tells us how does the virus affect the human body, right? But before we just jump into like a gene expression data, we actually wanted to teach them how are these count matrices generated. And the way they're generated is actually very simple, right? You just have these reads, which then you have to align to the genome and then see if they fall within a coding region. And we actually have them do this, right? Because we have the local alignment algorithm from the second exercise. And the very nice thing about this SARS-CoV-2 virus is that it's very small. It's 30,000 bases. And this is all stuff that can run on a laptop, right? So this is very nice. Then we have them run through this single-cell RNA-seq analysis. This is on real-world data, and then the typical steps, right? This is filtering, normalization, differential expression, and then we have gene enrichment analysis. And then as a bonus, they can also identify clustering or visualizing with TSNE and UMAP. Now, this might be a lot, but really this just goes through like a SCAMPI tutorial. So it's just to get a feel for the thing. Okay. So these are the assignments. Of course, we ask the students after every year that we do this, what we can improve, how much they like it. And in general, the students are very happy, right? So they find the exercises very interesting. Maybe they don't like the last exercise that much, but okay, can't always be happy. We also ask them how difficult they are. And assignment 3 is the hardest, right? And this is fine. Assignment 3 is the most coding heavy, and this one really requires a bit more programming knowledge, because if you sort of mess up, it can really take a long time. And in general, they don't find it easy, right? And this is good for us. I mean, we like that. We don't want this course to be that easy. We want it to be somewhat challenging and engaging, so we're happy with this. It also takes the third, how long does it take, right? It takes the students longer for the third exercise, which makes sense because it's harder. And then the first exercise as well, maybe because we're just throwing them in the deep end, right? Here's some FASTA file, figure out how to work with it, right? Okay, so I'm just going to conclude here now. So these exercises and these assignments are all publicly available. They're all on GitHub. So the link's down there. All of this is set up so it can be immediately run with GitHub Classroom. So these are just the instructions. We also have instructor notes if anybody is interested in trying to use this for their own courses. There are instructor notes on how to set this up, the GitHub Classroom and all that stuff. We also have solutions, but obviously we don't make them public because we are still trying to run this same thing. Yeah, exactly, until COVID remains relevant. But feel free to reach out and we can give you access. Thank you.
Thank you very much, Pavlin. A series of questions here. I'm going to start with.
So these CS students, how much biology do they have before they go into this class?
None.
So, I mean, concepts like evolution, is it taught to them?
Yes. So in the lectures, the professors, they do talk about some of this stuff, right? Like the first lecture tells you about the DNA and some statistical properties and the central dogma of biology. We don't really go specifically into evolution, like how does it work and stuff like that. But we assume that high school did teach them something, right? So probably everyone has some notion of evolution, right? Yeah.
Yeah, so thanks for the talk and the detailed explanation of the project. I wanted to ask you about keeping it updated because, of course, this is about the pandemic. And as you said, it was especially interesting when they could see in the news that the same thing happened. But COVID is still there and still infecting people. Have you done any updates with any new research paper or any new data, or are you planning to do that in the future, or even changing it from COVID to something else?
Okay, so until there's a new pandemic, hopefully not in the near future, we don't plan on changing it, right? Unless there's something that will be interesting and relevant. COVID is very nice because it's small and it can run on their laptops. So these algorithms are very manageable, right? We do update the exercises every year to as much as we can, right? So when the pandemic was going on, there was a lot of sequencing going on, and there was a lot of, for instance, when we were doing the variant exercises, there was an abundance of these sequences that we could get from our National Institute of Public Health or whatever it is. But now the sequencing has sort of stopped, right? Because there's no more pandemic. So we're sort of stuck where we are now, right? So we do update it as much as we can, but we feel like the core exercises are done. Okay.
Yeah, thanks. Have you seen any drop-off in interest as the pandemic kind of moves out of the news and is not perhaps so shiny and exciting as it was at the beginning of this?
So no, I think so. This is the only bioinformatics course that our faculty offers, and the students in general are very interested in bioinformatics. So the part that it's COVID related is just like an extra bonus, and maybe it was a bit more motivating at the start. But if anything, sort of the demographic that has changed. Before, the introduction to bioinformatics course was sort of easy-ish, and then a lot of people, first years we saw a lot of drop-off because they saw, oh, now this is hard. And then now we've got the better students who are more interested in it and who want to actually apply themselves and do it. So the attendance has not dropped off at all. So
It's good momentum, actually. Sounds like you're building momentum in the course.
Yeah.
Thank you. Please join me in thanking Pavlin, and we'll switch speakers.